Okay, so we'll be talking about neurotransmitters, the chemical messengers of our nervous system. And if you haven't already done so, you should click on this icon to download the student notes for this presentation. The student notes were not included in your main Unit 2 notes packet, so you will need this in order to proceed. Okay, so let's go over the basics of neurotransmitters. What are they? Well, they are the chemical messengers of the nervous system, like hormones are the chemical messengers of the endocrine system. They're actually produced in the soma of neurons, the cell body, and they travel down the axon, and then they are stored in the axon terminals in vesicles or sacs. So the neurotransmitters get made in the soma, they get put into these sacs, and they travel down that length of that axon and then get stored in the axon terminals. But where do they actually work? Because when they sit in the axon terminals, they're not really doing anything. Well, when there's an action potential, they are released into the synapse, and then they move across the synapse, and they work on the dendritic membrane of that postsynaptic neuron, opening up sodium gates. Okay. So here we have a drawing, and you can see the pre and the postsynaptic neuron, the synapse is in between them. It's also called the synaptic cleft or synaptic gap. It's all the same thing. There are your neurotransmitters stored in the axon terminal. You can't see them. Those neurotransmitters are inside of vesicles. And when there is an action potential, they're released into that synapse. And you can see on the postsynaptic neuron in that dendritic membrane, that's where those sodium gates are that they would adhere to or attach to. I'm going to show you this video here, and it's a quick one. I mean, it's about how neurotransmission works, and it gives you some context because we've already seen some of these diagrams before.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, so a little bit of context for what these neurotransmitters do, where they're stored, and now we're going to move into the actual types of neurotransmitters. And there are two types of neurotransmitters. There are those neurotransmitters that excite or create or generate an action potential on the postsynaptic neuron, or there are neurons that actually inhibit the next neuron in the chain from firing, and so they actually decrease the likelihood of an action potential. So we call these neurotransmitters excitatory and inhibitory neurotransmitters. Let me move my picture here. So excitatory neurotransmitters make the postsynaptic neuron generate an action potential. They increase the flow of the ions, those would be the sodium ions, right, into that postsynaptic neuron. They make it easy to depolarize. Remember, depolarize is that rising phase on the action potential chart that we went over. So they increase and make it easier for that membrane on the postsynaptic neuron to depolarize, allowing sodium to flow in, generating an action potential. And of course, they open sodium channels on that postsynaptic neuron. So it's all about opening these sodium channels or sodium gates that excitatory neurotransmitters do. Now, inhibitory neurotransmitters are the other way around. They prevent the postsynaptic neuron from generating an action potential. They make it difficult to depolarize, and they, instead of opening up sodium channels, they're actually opening up potassium channels. But remember, potassium is going to get brought back into the cell through the sodium potassium pump, that active pump that we talked about. So why would you need, I mean, you can imagine why you need an excitatory neurotransmitter. I touch something and that neural transmission takes a signal. It's an excitatory neurotransmission. It generates action potentials, and I get the signal to my brain that I touch something. I can feel cold, hot, pressure, warmth. Actually, I don't feel hot. We feel cold, pain, pressure, and warmth. Okay, so now why do I need to have an inhibitory neurotransmitter? Well, just imagine this. If a information flow comes in, at some point it has to stop, right? At some point the strength of the stimulus will stop. And not only that, but if I'm moving my arm, right? Am I moving it? At some point I've gotten to where I need to be. So I should stop that movement. I need an inhibitory neurotransmitter to make a signal that says stop. And I'll give you a third example. And the third example is, when you get really angry or upset, or you have a really high emotion, well, those are excitatory neurotransmitters sending information through your sympathetic nervous system. At some point, though, your parasympathetic has to take over and your body relaxes. It comes back to a homeostatic balance. And it can only do that because of these inhibitory neurotransmitters that stop the messages from reaching the organs, glands, muscles, and brain. Okay, today we're going to talk about the major neurotransmitters. I'm going to turn off my camera here. So the major neurotransmitters that we're going to talk about are adrenaline, which is also known as epinephrine, noradrenaline, also known as norepinephrine, dopamine, serotonin, GABA, acetylcholine, glutamate, and endorphins. And you have a chart that you should be taking notes on with this. And what we want to take notes on are some of the information that we see below the chemical structures of these neurotransmitters. For those of you in biology, AP Bio, you might actually have studied some of the chemical structures in here. So we'll start with adrenaline. You've probably all heard about adrenaline. It's that fight or flight neurotransmitter. It's actually also a hormone. So if you heard me talk about it in the endocrine system presentation, it is a hormone as well, but it's also produced in the nervous system. It is produced when we are responding to a stressful event. So it increases heart rate and blood flow, and it leads to like a physical boost and a heightened sense of awareness. So super aware of all the things going on. Okay? That is epinephrine. Norepinephrine is more about our attention. So the key word to write down here is attention. Okay? It is still involved with the fight and flight response. So it helps to contract blood vessels, which will, in effect, increase blood flow. So again, you're getting energy. So epinephrine and norepinephrine are two neurotransmitters. Let's move on to dopamine. And dopamine is our pleasure transmitter. It gives us feelings of pleasure, and it's also responsible for addictions. It's also responsible for movement and motivation. So a lot of different things. And dopamine sits all over our nervous system. So if it's in our muscles, it's responsible for movement. If it's in our central nervous system, it's more responsible for addictions and feelings of pleasure and motivation. We know that people repeat behaviors that lead to a dopamine release. So some people say when they eat chocolate, they get a dopamine release. Well, they'll be more likely to eat that chocolate again. Or if you run, you get that runner's high, you will get a dopamine release and it will make you want to run again. Okay? So dopamine is a pleasure neurotransmitter, but it's also about movement. So it's certainly when it's sitting in our somatic nervous system, and it's part of our efferent, our motor pathways, it's also part of movement. Serotonin is our mood transmitter, and it's the one most implicated in depression because it contributes to our well-being and our sense of happiness. It also helps to regulate our sleep cycle and our digestive system. And we know that serotonin levels are affected by exercise and exposure to light. So we see increased levels of serotonin when we exercise, just like we would see increased levels of dopamine if exercise is pleasurable for us. So just to review, we have the epinephrine, which is about the fight or flight, norepinephrine, It's our concentration neurotransmitter. Dopamine is our pleasure neurotransmitter, and our movement. I think you need to make sure you note that. And serotonin's about mood. Let's move down to GABA. GABA's the calming neurotransmitter. It's actually an inhibitory neurotransmitter. It's our body's main inhibitory, and it calms the firing of nerves in our central nervous system. We know that people with high levels of GABA, in other words, people who are relaxed, can actually improve their focus. We know that low levels of GABA, meaning that it's not producing a calming effect. Remember, it's the calming neurotransmitter. So if you don't have enough of it, you won't be calm, you'll be anxious. We also know GABA does play some role, just like dopamine, in motor control. Let's move on to acetylcholine. Sometimes you hear it called acetylcholine. It's our learning transmitter. It's all about learning and memory. So it's involved in a lot of that stuff that is in our frontal lobe. So learning, memory. It also does work with muscle action throughout the body. So we see, as you can see, there's a lot of overlap in neurotransmitters, and they exist all throughout the nervous system. So depending upon where they are, their action may change. So certainly in the frontal lobe, acetylcholine or acetylcholine is involved with thought, learning, and memory. But when it's in the efferent pathways, just like dopamine, it will affect muscle action in the body. Let's move on to glutamate. So while GABA is our body's main inhibitory neurotransmitter, glutamate is our body's main excitatory neurotransmitter. In fact, it's the neurotransmitter we have the most of in our body. So again, it's the most common brain neurotransmitter. It's also involved with learning and memory, and it helps to develop and create nerve contacts. In other words, where the two nerves meet, the junctures of those neurons, the synapses. So it helps to facilitate or helps to grow synapses between multiple neurons. So it's a really important neurotransmitter when it comes to the excitatory piece of our neurotransmission system. Finally, let's move on to endorphins. So, we can get an endorphin high, and it will be released. Often endorphins are released during exercise. They're released really during any period of euphoria. So it could be exercise or some exciting event or sex, and it does produce a sense of well-being. But it also reduces pain. Okay? And we know for certain that endorphins are released, and it's responsible for some people being able to appear to be super strong during a stressful event. It's not that they're super strong, it's that they're actually working through tearing up their muscles because they're not feeling the pain because the endorphins are there. So here are our major neurotransmitters, and if you haven't been able to take the notes as quickly as I've been speaking, please pause here before we move on and talk a little bit more about what happens when we have too much or too little of each of these. Okay, so here we are with the same neurotransmitters, and I really want to now go through what happens when you have too much or too little. In other words, what happens when there's an imbalance, a neurotransmitter imbalance. Neurotransmitter imbalances are implicated in many neurological disorders. We know with adrenaline that's our fight or flight neurotransmitter. When you have too much adrenaline, you can actually feel dizzy. So it causes a lightheaded feeling. With norepinephrine, since it's our concentration neurotransmitter, if you have too much of it, you'll actually be so hyper-focused, you're aware of everything, you might become anxious. But we also know that when you don't have enough of it, that it is implicated in obsessive compulsive disorders, lack of focus, and even a depressed mood. So not so much depression, but a low level of a mood, not quite depression. With dopamine, our pleasure neurotransmitter, remember, it's one that is involved with movement and motivation. It appears that too much dopamine is associated with hallucinations. And hallucinations are sensory perceptions, or like perceptions of things that are there, whether it's visual or auditory or tactile, without there being any appropriate sensory input. So it's seeing something that's not there, hearing something that didn't make a sound, feeling something on your skin that is not present. So hallucinations that like we see in schizophrenia are, we know, in part due to too much dopamine. Now, again, since it's involved with muscle movement and coordination of muscle movement, if you don't have enough dopamine, you won't be able to move your muscles in a fluid and smooth manner, and you might suffer from tremors, as we see in Parkinson's patients. So too much dopamine, hallucinations, schizophrenia. Too little dopamine, tremors, Parkinson's. Okay? Now, serotonin is involved with mood and sleep regulation. If you have too much serotonin, you might end up with certain sleep disorders. You actually could have hypersomnia. In other words, you sleep all the time, or you could have insomnia where you cannot sleep at all. So it's kind of one of those neurotransmitters that you just need that right balance. But we know for sure that for most people that suffer from depression, there is an imbalance of serotonin and there's too little of it. So depression is in part caused by too little serotonin. We also see that patients who have obsessive compulsive disorders don't have adequate levels of serotonin either and can benefit from antidepressants. So GABA is our calming neurotransmitter. And since it's our calming one, it's inhibitory. If you have too much of it, it actually will cause you to feel sedated or be sedated, so go to sleep. And again, since it's our calming neurotransmitter, if we don't have enough of it, our brain could be overactive, inducing seizures and tremors and insomnia. So it's really important to have those levels of GABA to keep our body or maintain homeostasis or return our body to homeostasis. With acetylcholine or acetylcholine, too much we see, because it is involved with muscle movement. So when you get too much acetylcholine in your body, you can see muscle convulsions. It's really rare to have that, but there are certain types of poisons that actually can produce too much acetylcholine and give you muscle convulsions. Now, too little acetylcholine, because it's involved with memory and learning, would actually impair your memory. And we see with people who have Alzheimer's that they have too few neurons that are producing acetylcholine. So they're not able to create memories, pull things from memory. We also see that in the muscle regions where acetylcholine is present, that too little acetylcholine can cause paralysis in the muscles. So too much is overactive muscles, right? Muscle convulsions. Too little, paralysis in the muscles. So move on to glutamate. We're only going to look at glutamate from the fact that if you have too much, we know since it's an excitatory neurotransmitter, too much could produce things like migraines or even seizures. Right? So certainly when you have an excitatory neurotransmitter like glutamate, if there's nothing modulating that, if you have too much of it, you're going to see overactivity in the central nervous system, which can produce those migraines and seizures. Endorphins, that's our euphoria neurotransmitter. It's also the one that helps to stop pain. Too much, though, seems like, okay, well, can you have too much euphoria? Sure. You can get really edgy. You can be really quick to respond with that fight or flight reflex. For example, you might have just come out of a game that you've been watching, a homecoming game, and you're really excited your team won and you're on an endorphin high, and then somebody accidentally touches your shoulder in the parking lot and you get really angry at them. You turn around like ready to fight them. That's those endorphins going. It's not that they're touching you really made you angry, but you were already on the edge. Okay, so that was a quick to fight or flight response. Now, also with endorphins, we know with too little endorphins, we have pain and we can see sadness. And there certainly are disorders where people feel body pain, okay, all over. And it may be in part due to too few endorphins. Okay, so on your chart, you actually will see on the rightmost column things called agonists and antagonists. These are drugs that act like or block the actions of these neurotransmitters. So while I did write these in for you, you are expected to know them, and they are really interesting. So I'm just going to highlight a few while I'm on this slide. We do know that antidepressants are like serotonin, so they can help to cure, or at least partly help to cure, depression. So that's just one. I also bring up something like MSG, monosodium glutamate, in food. That actually acts like glutamate. So people who have migraines have to avoid monosodium glutamate in food. We know that when people have anxiety, we often have to give them a GABA agonists because they don't have enough GABA to feel calm. So there's a lot of drugs, and we're going to go over agonists and antagonists in our next slides. But please, as we go over them, take a look at that column. So what is an agonist? These are an antagonist. These are substances that act as neurotransmitters. They could be drugs that are manufactured by large drug companies, or they could be found naturally in the environment. So, for example, botulinum toxin is found naturally in the environment. It's also produced in a lab, but we know when people get botulinum poisoning, they seize up, they can't move their muscles. Well, botulism or botulinum toxin is produced in the labs, and it's actually used to treat movement disorders. You can inject it into muscles and paralyze the muscle to stop a tremor or stop a movement. It's also used for in the plastic surgery field to stop a muscle from moving to show that there's, or at least produce the effect of having few wrinkles. Okay, so agonists are drugs which imitate the action of a neurotransmitter in the brain, or it blocks its reuptake. And we are going to go over what reuptake is, but do write that down. An agonist is a drug which imitates the action of neurotransmitters or blocks its reuptake. An antagonist is a drug that blocks neurotransmitters. And I'd like you to think of neurotransmitters and the sodium channels or sodium gates on that postsynaptic neuron. You have the drugs or the neurotransmitters that are like keys, and then you have the gates, which are like locks, and one fits into the other. And if it's an agonist, it will fit in and do exactly what that neurotransmitter was supposed to do. If it's an antagonist, it fits in, but it's going to block that neurotransmitter from doing its action. So here is a nice graphic which will show you that. So note how an agonist, okay, in this case it's a drug, fits like a key into a lock. So if that agonist happens to be binding to a receptor site that is meant for serotonin, okay, then you would see that it would link in and actually produce the same effect as serotonin. But an antagonist, while it fits into the lock, it doesn't fit well enough to open it or to turn it. I use the metaphor of turning it, but it really does not open it. So it actually blocks then the neurotransmitter that's meant to go in there from working. So it's similar to when you put the wrong key. So that antagonist is similar when you put the wrong key into the lock of your front door or your locker, or the key seems to fit, it goes in, but it won't turn. Okay, it won't open the latch. So when do we give an agonist and when do we give an antagonist? So an agonist, and of course we're talking now if you're prescribing drugs, an agonist is given when there's too little of a neurotransmitter, because again, it will produce the same effect of a neurotransmitter. But an antagonist is given when there's too much of a neurotransmitter. And when there's too much of a neurotransmitter and you want to stop that neurotransmitter from flowing through, you have to give an antagonist, a drug that will just block the action of the neurotransmitter. So if you look back here, we've talked about this agonist, and we say it imitates it, or it can block its reuptake. So I'm going to go back to the previous slide. This is an agonist imitating a neurotransmitter, but an agonist works in a second way. An agonist can work by blocking something called reuptake. Reuptake is the absorption by the presynaptic neuron ending of the neurotransmitter that it's secreted into the synapse. So neurotransmitters, as you saw in that first video, get secreted into the synapse. And you also saw in that first video that they get reabsorbed into the sending or the presynaptic neuron. Well, an agonist will block that process. And that means that the neurotransmitter sits in that synapse a bit longer, giving it a chance to do its job. So, for example, people who are treated with things like Prozac for depression, it's an antidepressant. It's a serotonin agonist. It produces the same effects as serotonin, but it doesn't produce the effects of serotonin by fitting like a key into a lock. It produces the effects of serotonin by blocking these reuptake channels, these reuptake pumps. And it keeps the natural forming serotonin here in the synapse longer and lets high levels of serotonin build up to increase the likelihood that the next neuron in the chain will fire and a sense of well-being will be achieved. I'm going to go ahead again. This is that video you saw earlier, and we're going to fast forward it to the 1 minute and 2 second mark. The term reuptake is not used in the video, but when you see the neurotransmitters transported back into the sending neuron, that is reuptake.
Communication.
Just going to fast forward. Okay, here we go.
Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, so you saw when those neurons got reabsorbed into the presynaptic neuron, and let's just be clear that the presynaptic neuron, what you're seeing there is the axon terminal there. And what you're seeing there is in the blue is the synapse. And those receptor sites are on the dendritic membrane of the postsynaptic neuron. And remember back to our neuron diagram that the dendrites were those bushy extensions of a neuron that receive information from other neurons. Okay, so that is neurotransmitters, and we will have some practice with that. And I hope that you got some interesting information. And please definitely look at that third column on your chart, because that third column has a lot of good information about the agonists and antagonists, very specific ones to treat disorders.